In this video, we're going to talk about how to approach A-level exam questions, and in particular, how to use some of the information that we're provided with in order to build a suitable model answer. Now, this question is obviously on the topic of photosynthesis, and it gives us a paragraph or a passage that it does ask us to first read. So what I would do is to take a highlighter pen and just highlight the important bits or the important terms or key terms within this passage before we actually start to think about the answer. So it says, the living state requires a constant input of energy, and the most fundamental difference between animals and plants is the way that they actually obtain this energy. Now we know animals take in food, complex organic material, and release chemical energy during aerobic respiration. So we know animals are heterotrophs. We know they consume normally through holozoic digestion, complex organic material like glucose, amino acids, etc. And we know that all or certainly most of the cells in animals respire glucose aerobically inside their mitochondria. And we know that at the end of aerobic respiration, lots of ATP is being produced. And one tip that I would recommend is, if you do have a question on either respiration or photosynthesis or both, is to just write out the simple chemical equation for these processes. So we can do this in the margin. We know aerobic respiration is the breakdown or oxidation of glucose, which is a monosaccharide. We know it involves 6 molecules of oxygen per molecule of glucose, and this occurs inside the mitochondria. We also know that 6 molecules of carbon dioxide are given off per molecule of glucose, and they come from the mitochondria as well, and in the matrix from the link reaction in the Krebs cycle. We know that 6 molecules of water are being produced, and we know that water is actually formed from the oxygen at the end of the electron transport chain, because oxygen is the final electron acceptor, and it picks up protons and electrons. It pulls the electrons through the ETC, and when they're combined, we get water. And we know there's 38 theoretical molecules of ATP being formed per molecule of glucose. So we can just write this in the margin at the top of the question. Now the question then goes on to say, green plants absorb light energy, and we know that comes from sunlight, and they convert this light energy into chemical energy in the process of photosynthesis. So again, in the margin, we can just simply write down the chemical equation for photosynthesis, and we know this time plants that do photosynthesize, they take in carbon dioxide from the atmosphere, so we could put here from the air, and we know they obtain water from the soil, and they absorb that from the soil water through the root hair cells. So CO2 and water are the raw ingredients for photosynthesis. Now we know light energy is required, and that's absorbed by the photosynthetic pigments in the thylakoid membranes of the chloroplasts. Now we know the end product of photosynthesis inside the chloroplasts is the production of glucose from the Calvin cycle. And a waste product is molecular oxygen, which we know is given off through photolysis, which is the light-activated splitting of water. So we're going to put these in the margin, and this is going to help us approach the question and think about what we need to write about. So question A says, describe how plants absorb light energy from the sun, and then how they use this energy to produce useful substances in the light-dependent stage of photosynthesis. So we need to think, first off, how plants actually absorb the light energy. So we might want to start to talk about photosynthetic pigments, where they're located, for example in the thylakoid membranes, which are inside the chloroplasts, inside plant cells that may photosynthesize. And then it says, describe how they use this energy that's just been absorbed, the light energy, to produce useful substances. Now we know from the light dependent stage, which is really the first stage of photosynthesis that we describe, The two useful substances are ATP and reduced NADP. So at the end of the light dependent stage, that's actually going to involve photosystem 1 and photosystem 2, the two different photosystems with their antenna complexes of accessory pigments. And we know that at the reaction center is 2 molecules of chlorophyll A. We're going to get ATP being formed from ATP synthase, and we're going to get NADP being reduced. And we know NADP, when it's reduced, it has acted as the final electron acceptor. Now, both of these molecules are going to be needed in the Calvin cycle in the stroma of the chloroplast in order to be able to make triose phosphate, which is then used to make glucose. So the mark scheme for this particular question then, we can just tick our way through it. First mark is, we know plant cells have chloroplasts because the question is asking us how plants absorb light energy from the sun, the first part of the question. We then know inside the chloroplasts are thylakoid membranes, and they're arranged into granule stacks. And it's the thylakoid membranes inside the chloroplasts that contain photosystem 2, which we normally draw on the left, and photosystem 1, which we normally draw on the right. Now, in between these two photosystems, with PS2 on the left and PS1 on the right, is the electron transport chain. Now, the third point is to then talk about the accessory pigments and what their role is. So we know the accessory pigments absorb a wide range of different wavelengths of light, photons of light, and pass the excitation energy down to the 2 chlorophyll A molecules in the reaction center of both PS2 and PS1. So we've talked about the different pigments, accessory pigments like beta-carotene, xanthophyll, etc., called the carotenoids. Chlorophyll B is an accessory pigment, and we've also talked about the chlorophyll A, which is the primary photosynthetic pigment in the reaction center of both PS1 and PS2. Now we know that 2 high energy electrons get released from the reaction center of PS2, and they pass down the electron transport chain that connects PS2 on the left and PS1 on the right, and this occurs within the thylakoid membrane. So the excitation energy being passed down to chlorophyll A in the reaction center energizes 2 electrons, which get released from the reaction center and therefore from photosystem 2. They get picked up by the primary electron acceptor that passes the 2 high energy electrons down the ETC, and we know as they pass down the ETC, these 2 electrons actually lose energy, and that energy is activated or is used to activate a single proton pump within the electron transport chain between PS2 and PS1. And that proton pump, when it's activated, pumps in protons into the thylakoid disc. The thylakoid disc becomes very acidic because of the accumulation of protons, and it helps to establish the electrochemical gradient. Now, this is a little bit similar to what happens inside the mitochondria in terms of proton pumps being activated and protons being pumped into the intermembrane space. But here, the high energy electrons, they actually come from the reaction center of PS2 on the left. Now, hydrogen ions, the only way they can diffuse back into the stroma from the thylakoid disc lumen is through the Stork particle. And we know protons will move down their electrochemical gradient from the lumen of the thylakoid disc back into the stroma through the Stork particle. And excuse me, the catalytic domain of the Stork particle is ATP synthase or ATP synthetase. Now, these protons are full of potential energy, and as they do move back through the Stork particle, it activates ATP synthase, and it allows the formation of ATP from ADP and inorganic phosphate. Now, the high energy electrons from photosystem 1, from that reaction center, when they get released, they do not pass down the electron transport chain, but instead they get combined with protons and oxidized NADP, which results in the formation of reduced NADP, and we call that NADPH2. Now, oxidized NADP is the final electron acceptor in this process called non-cyclic photophosphorylation. Now, the electrons that did move down to ETC, as they lose energy, they become low energy, and they actually replace the two electrons that were energized and released from the reaction center of PS1 on the right hand side. So we've talked about how ATP is formed. We've talked about how reduced NADPH2 is formed. And these two products, like we said at the start of the slide, are needed in the stroma in the Calvin cycle to allow the formation of triose phosphate and therefore glucose. Now this question here does give us a diagram that we're going to use and help us to construct an answer. And it tells us the diagram shows part of the light independent reactions of photosynthesis. Now we know this really means the Calvin cycle, and we know that occurs in the stroma of the chloroplast. So we can start to use the information and work out what's happening before we even approach the questions beneath. Now we know carbon dioxide is a gas and it's in the stroma of the chloroplast, and we know that's actually combined with ribulose bisphosphate, which is a 5-carbon sugar molecule called pentose sugar, and that will form 2 molecules of glycerate 3-phosphate. Now this reaction, we can annotate the question itself. It's actually catalyzed by an enzyme called Ribisco, and that stands for ribulose bisphosphate carboxylase oxygenase. So this reaction here, because it's adding carbon dioxide or fixing carbon dioxide onto the 5-carbon ribulose bisphosphate, is a carboxylase reaction. So we can put here carboxylase. Now you might have seen decarboxylase before during aerobic respiration when you talked about carbon dioxide being released or removed from a substrate. Well, here it's the opposite. It's being fixed onto a substrate. So we call it a carboxylation reaction by Ribisco, catalyzed by Ribisco. Now that actually forms an unstable six-carbon molecule, and because it is unstable, it then splits into 2 3-carbon molecules called glycerate 3-phosphate. Now we know glycerate 3-phosphate is a 3-carbon and it has a phosphate group attached to it. Now each glycerate 3-phosphate in the stroma of the chloroplast will undergo a reduction reaction. Now the hydrogen comes from reduced NADP, and we know this is the end product of the light dependent stage from non-cyclic photophosphorylation. So sometimes you see this as NADPH2. So this hydrogen here is going to be used in the reduction of G3P to triose phosphate. And the energy that drives this reduction reaction comes from the hydrolysis of ATP. So this is an enzyme catalyzed reaction. G3P is reduced using the hydrogen from reduced NADP. As the hydrogen is removed from reduced NADP, obviously oxidized NADP is reformed. So we can put here reformed. And that's needed because that actually allows the continued reactions of non-cyclic photophosphorylation. So without any oxidized NADP, we can't have continued light-dependent stages. Now we know ATP, when that's hydrolyzed, that forms ADP and inorganic phosphate, and it releases 30.1 or 30.6, kilojoules per mole of energy. 30.6 kilojoules per mole of energy that drives this reduction reaction. So reduced NADP and ATP are both needed to reduce glycerate 3-phosphate into triose phosphate. So if we get 2 molecules of G3P, we put here 2 G3P, we're going to end up with 2 molecules of TP, as long as there is reduced NADP and ATP made from the light dependent stage. Now, this is just annotating the information we're given. It doesn't take long if we've learnt the information to do that. Now, one thing we could also add here is that we know the Calvin cycle must go around 6 times in order to make 1 glucose molecule. So if we have 6 Calvin cycles, we'd actually end up with 12 triose phosphate, because 2 TP come from 1 Calvin cycle. So we've got 6 cycles, we get 12 TP. Now we know 2 TP, because they're triose phosphates, are needed. So we could say 2 TP are needed to make 1 glucose, because 2 triose phosphates make 1 hexose. Now the remaining 10 TP, actually that's 10 triose phosphates, so it's 30 carbons. They're actually used to regenerate 6 ribulose bisphosphate. Now because these are 5 carbons, if you've got 6 of them, we get 30 carbons in total. So 10 triose phosphates, which is 30 carbons, are needed to make 6 ribulose bisphosphate molecules, which again is 30 carbons, and that also does require ATP. But this time the phosphate group is actually needed to help make ribulose bisphosphate. Bis means 2. So that's the basic gist of the question and the information that we're given. So the first question, A number 1, says how many carbon atoms are there in 1 molecule of G3P? Well, we know straight away that is 3. Then it says, what is the function of each of the following in the reactions shown in the diagram? The reduced NADP. Well, we know that hydrogen is actually needed to help reduce glycerate 3-phosphate into triose phosphate. So reduced NADP, and you can write it in this format, when that's oxidized, we know oxidation is loss of hydrogen. The hydrogen is actually released, and that gives off 2 high energy electrons, and it's used, or they're used, to reduce G3P into triose phosphate. So this is actually a redox reaction where the reduced NADP is being oxidized and the G3P is being reduced using that hydrogen or those electrons. Now the ATP we know is hydrolyzed by an enzyme, and that releases ADP and inorganic phosphate, as well as, like we said earlier, a small amount of energy or a small packet of energy, and it's that energy that is needed to drive the enzymatic reduction of G3P to triose phosphate. So without ATP, we can't get G3P being reduced to TP because it does require this small packet of energy. So in question B, we're going to build on some of the information that we've just put into the exam question. Now this question says, describe how the products of the light-dependent stage of photosynthesis are used in the Calvin cycle and how carbohydrate is synthesized as a result of the cycle. So there's actually 2 parts to this question. The first part of the question is asking how the products of the light-dependent stage are used in the Calvin cycle, which is what we've just talked about. And the second part of the question is actually asking how carbohydrate is synthesized as a result of the cycle. So we know the first part of the answer then is to talk about glycerate 3-phosphate being reduced to form triose phosphate. That's the first mark point. We know that uses the 2 high energy electrons that are carried into the stroma and into this reaction by reduced NADP or NADPH2, as you may write it. That's the second mark point. We know ATP hydrolysis into ADP and inorganic phosphate is needed to supply the small packet of energy needed to drive that reduction reaction. But now we need to talk about the second part of the question then. So how is carbohydrate synthesized as a result of the cycle? Well, we know triose phosphate is used to regenerate ribulose bisphosphate using some ATP in the stroma. Now that's really important because ribulose bisphosphate is a pentose sugar, so therefore technically it is a carbohydrate. It's a simple sugar, and it is needed to allow the continued cycling of the Calvin cycle, to allow it to keep going round time after time, because it is one of the starting molecules for the Calvin cycle. Now we also know that triose phosphate is used in order to make glucose, and we know it's one glucose molecule being made from every 2 triose phosphate molecules, and that also consumes some ATP. So not only is triose phosphate needed to regenerate the ribulose bisphosphate, it's also being used, 2 of them, to make one glucose molecule. Now we know in plants, because plants can't take in ready-made organic material, they have to make all their own complex organic material from scratch. Now we know glucose is the starting molecule for many different organic molecules inside plant cells. So this glucose can often be used to synthesize other carbohydrates like sucrose, and we know sucrose is really important in plants because it's translocated away from the leaves up or down the sieve tubes in the phloem. We know glucose can be used to make starch, and this is a storage structure for chemical energy, and it's made of amylose and amylopectin. And we also know glucose is needed, beta glucose, to make cellulose. Now cellulose is very important in plant cell walls. So triose phosphate is needed to regenerate ribulose bisphosphate, marking point number 4. It's also needed to make glucose, point number 5, and the glucose is then needed to make other organic molecules like sucrose, starch, cellulose, and even molecules like amino acids and chlorophyll, which are organic-based molecules. Now the next question says, describe the role of water in the light-dependent stage of photosynthesis. So we have to know about photolysis, and we know photolysis can only occur in the light. So the name photolysis, suggests the splitting of water activated by light. So here we can start to talk about water is split by photons of light in photolysis, and this happens at photosystem 2 in the thylakoid membrane on the left side, and water then gives off protons or H+ ions, 2 low energy electrons, and also oxygen gas. Now we know oxygen gas is a waste product. This is actually where the oxygen gas does come from, the splitting of water, called photolysis. And in the exam you do actually need to write down these 3 products of photolysis. It's not good enough just to say it's the splitting of water by light. So protons, electrons, and oxygen gas are released. Now we know the role of the low energy electrons and what they're needed for. So they replace the high energy electrons, the 2 high energy electrons released from the reaction center of photosystem 2 on the left. Now we know the reaction center is made of 2 chlorophyll A molecules, and 2 high energy electrons get released after they've been energized from reaction center of PS2. So in order for continued electron transport chain reactions to occur and electrons to continuously move down the electron transport chain, these 2 high energy electrons must be replaced by low energy electrons released from the photolysis of water, and that occurs at photosystem 2. There's actually an enzyme that's activated by light that helps to catalyze the splitting of water into protons, electrons, and oxygen gas. So further building on what we've just written down then, the next question this time says, explain why the amount of G3P increases after a photosynthesizing plant has been in darkness for a short time. So we can take our highlighter pen, we can highlight G3P increases after the plant's been left in darkness for a short time. And there's 2 parts to this answer. So 2 markers, we need 2 separate sentences to get the 2 marks. Now, the first part of the answer then we can think about, well, we know G3P is still synthesized in the dark. This does not require light, and it does not require products from the light-dependent stage. Now this is because the fixation of carbon dioxide with ribulose bisphosphate is catalyzed by Rubisco, and this does not need ATP and it does not need reduced NADP. So this occurs in the stroma of the chloroplast, and it allows the 5-carbon ribulose bisphosphate to be combined or fixated with the 1-carbon carbon dioxide. We get the unstable 6-carbon compound, which then splits into 2 G3P. So glycerate 3-phosphate can still be made in the dark as long as there is Rubisco, ribulose bisphosphate, and carbon dioxide available. So that's the first mark point. Now the next mark point is to think about why GP increases then. Why does it not just stay constant? Well, we know GP is not being reduced to make triose phosphate, and that's because, like we just said earlier, there's no ATP and there's no reduced NADP being made from the light dependent stage because it's in the darkness here. So if there's no ATP and there's no reduced NADP, G3P cannot be reduced to triose phosphate. So it's still being made in the dark for a short amount of time, and it's not being used, therefore its concentration increases in the stroma of the chloroplast. So that's the 2 marks there. Right, the next question is the 6th marker, and it says describe the similarities between photosynthesis and respiration. So this time we don't need differences, of which there are many. We actually want similarities, but we can actually get the similarities by thinking about the differences. So first thing we want to think about then is we know both photosynthesis and respiration involve the transfer of energy or the conversion of energy from one form to another. So for example, in photosynthesis we've got light energy being converted to chemical energy, and in respiration we've got chemical energy being converted into chemical energy but in a different type of molecule, i.e. ATP. The next similarity is that they both, respiration and photosynthesis, both use and produce ATP. Now we know aerobic respiration makes 38 ATP theoretically inside the mitochondria, but also respiration does use up some ATP, and that's the investment phase in glycolysis, because we know that uses 2 ATP and it converts glucose into hexose bisphosphate, that 6-carbon sugar with 2 phosphate groups added to it. So they both use and produce ATP. Now in photosynthesis we know ATP is produced from non-cyclic photophosphorylation, and that occurs in the thylakoid membrane. But we know ATP is actually used up during the Calvin cycle to make G3P and to also convert TP back into or regenerate into ribulose bisphosphate. So the second mark point is that they both use and produce ATP. And you might want to give some examples there just to back up your statement. Now the third mark point then, similarities. In the inner mitochondrial membrane is an electron transport chain, and that's made of electron carriers and 3 proton pumps. And in the thylakoid membrane there is also an electron transport chain. Now as this question is asking for similarities, that's sufficient just to write that. If it was asking for differences, we could start to talk about the different numbers of proton pumps. So we know in the thylakoid membrane there's actually 1 proton pump between PS2 on the left and PS1 on the right, and we know in the inner mitochondrial membrane there's actually 3 proton pumps. But because we're talking about similarities here, it's sufficient just to say the electron transport chain is involved in both processes. Now the fourth mark point then out of 6, we know the inner mitochondrial membranes and the thylakoid membranes provide a large surface area for the production of ATP by phosphorylation of ADP. So we know there is ATP synthetase or ATP synthase as part of the stalk particle in both the inner mitochondrial membrane, where the electrochemical gradient is built up, and also in the thylakoid membrane, where the electrochemical gradient is built up between the stroma and the lumen of the thylakoid disc. So ATP synthase as part of the stalk particle is used to make ATP by phosphorylation of ADP and inorganic phosphate. Now we know, mark point number 5, respiration and photosynthesis both involve establishment of that electrochemical gradient, so that's a nice similarity. If it was a question about a difference, we could start to talk about the intermembrane space and the matrix, and the thylakoid lumen or thylakoid disc lumen and the stroma. But because it's a similarity, we can just talk about the electrochemical gradient, and that results in chemiosmosis, which results in phosphorylation of ADP to make ATP. Mark point number 6. Respiration involves a cycle of reactions called the Krebs cycle, and that occurs inside the matrix of the mitochondrion. Now, photosynthesis also has a cyclical set of enzyme reactions called the Calvin cycle, and that occurs in the stroma. Now, another similarity we could talk about is that we know coenzymes are involved, and we know coenzymes such as NAD or FAD in respiration, or NADP, if we're talking about photosynthesis, they become reduced when they bind hydrogen, and we know hydrogen contains electrons. If there's 2 hydrogen atoms, then there's 2 electrons. So these coenzymes can become reduced, and we also know that they give off their hydrogen or electrons, so they become oxidized. So both respiration and photosynthesis both involve redox reactions, which is oxidation and reduction occurring simultaneously. Now, one last point that we could also add is that we know these coenzymes are very, very important to allow the enzyme reactions that occur in both photosynthesis and respiration. So the catalyzed reactions, the enzyme reactions, will not occur unless these coenzymes are present at the same time, hence the name coenzyme. Now, you don't have to mention the differences here because we're not after a difference, we're after similarities, but just for reference again, it's NAD in respiration and FAD as well inside the mitochondria. It's involved in the Krebs cycle, and in photosynthesis it's NADP, and that is the final electron acceptor at the end of non-cyclic photophosphorylation. Now this is a slightly different style of question, and it's going to give us some data in the form of 2 different graphs, and we're going to utilize this data to be able to formulate a response and an answer. So the information in the text at the top of the question, it does say that red seaweeds are algae. Now algae are unicellular and sometimes multicellular organisms that can photosynthesize. They are autotrophic. They can make their own glucose through photosynthesis. So they do have photosynthetic pigments. And you can take your highlighter pen and you can sort of highlight the important bits of the question. And it does say that these red seaweeds, in addition to chlorophyll, they have a red pigment called phycoerythrin. So it does tell us, and we can jot this down as well, that these algae do photosynthesize, and they have both chlorophyll as a photosynthetic pigment and the other second pigment called phycoerythrin. So it has both of these pigments. It then tells us that green seaweeds do not contain phycoerythrin. Okay, so this one here at the top, this would be the red algae, and I can just put here the second type of algae we're going to talk about just has chlorophyll without any phycoerythrin, and that is the green algae. Then it says both phycoerythrin and chlorophyll absorb light energy, which can be used in photosynthesis. The graphs show the percentage of light of different wavelengths absorbed by seawater. So this is the graph at the top here. By chlorophyll and by phycoerythrin, which is the graph at the bottom. Then it says use the info from the graph or graphs to suggest why red seaweeds are found in deeper water, i.e. further down the shore, nearer to the water as it comes up the shoreline. But green seaweeds are not found further down the shore in deeper water. So when we start to look at the data, what we can do is you must look at the labels that were given to start with on any graph. Now the x-axis we know runs across the bottom of each graph here, and that tells us the wavelength of light, and it's exactly the same scale for these two graphs. So we can see here it starts at 400 nanometers and it ends at 700 nanometers, and that's the same for the graph on the bottom, 400 nanometers to 700 nanometers, and that's the wavelength of light. Now this is the visible part of the electromagnetic spectrum, and we know light of different wavelengths have different colours, and we're actually given the colours on the x-axis on the graph at the bottom of the question. So we know 400 nanometer light is violet light, 500 nanometer light is greenish light, 600 nanometer light is yellowish orange, and 700 nanometer light is red light. And we're going to use some of this information in the response in a second. Now, if we start to look at the absorption spectrum at the bottom, so the bottom graph, it gives us a dotted line that shows us how much light energy is being absorbed by phycoerythrin, this pigment, at the different wavelengths of light. And we can see by looking at these two crosses that most light energy is being absorbed in the blue-green part of the visible spectrum and the yellow part of the spectrum, because the curve here, the highest peaks on the curve correspond to around about 470, 480 nanometers and about 570, which is green and yellow light. So most light absorbed at these wavelengths by phycoerythrin, and that's going to be bluey green light and yellow light. What we can then do is correlate these peaks on the bottom graph, i.e. the wavelengths of light at which most light energy is absorbed for phycoerythrin, with how much light is being absorbed by the seawater. So we can see here, actually there's quite a low percentage of light being absorbed by the seawater, and that suggests that this light of these wavelengths can actually penetrate the water, and therefore it can be absorbed by the phycoerythrin pigment in the thylakoid membranes of the red algae. So above the graph above the absorption spectrum at the bottom shows us the least amount of light being absorbed at these wavelengths by water, because obviously this is towards the bottom of the curve, which means that these wavelengths of light can penetrate the water and therefore be absorbed by the pigments excuse me, in the cells of the algae underneath the water, which are, as we saw in the question, deeper down the shoreline in deeper water. Now when we look at chlorophyll, this dotted line on the bottom absorption spectrum, we can actually see that most light, the highest amount of light energy, is being absorbed in the blue part of the visible spectrum and the red part of the visible spectrum for chlorophyll. So we can see the curve, it peaks at this point here where the X is, and that shows the most amount of light energy being absorbed is blue light, and then it also peaks, but not quite to the same extent, in the red part of the visible spectrum. So this X shows us that excuse me, there's also light being absorbed in the red part of the spectrum by chlorophyll, the pigment. Now when we correlate these peaks where most light is being absorbed by chlorophyll in the red and the blue parts of the visible spectrum, we can see actually there's a higher level of these wavelengths of light being absorbed by the seawater, which means red and blue light don't penetrate the water quite as easily as green and yellow light. So chlorophyll if that was in the thylakoid membranes of the red algae, it would not be able to absorb the red and the blue light as easily, because the red and the blue light is being absorbed by the water, which means it can't penetrate through the water. So we're going to use these ideas in our response on the right hand side. And it says, use the info from the graph to suggest why red seaweeds are found in deeper water down the shore, but green seaweeds are not. So our first response is to state green and yellow light, approximately 500 to 600 nanometers. So we use data off the graph itself. Can penetrate water better than red and blue light, as less of the green and yellow light is being absorbed by the water. Now, as the red seaweeds have phycoerythrin, which absorbs green and yellow light to the greatest extent, the red seaweeds can photosynthesize better in deeper water. So the red seaweeds have phycoerythrin. We know these absorb quite a high level of both green and yellow light using the absorption spectrum at the bottom. So the red seaweeds can photosynthesize better in deeper water. Now, blue and red light, approximately 460 to 670 nanometers, penetrates water very poorly, because we showed at the top that it's actually absorbed to a higher extent here and here. So this light, blue and red light, is absorbed by chlorophyll. Green seaweeds cannot photosynthesize in deep water because they only have chlorophyll pigments and they do not have phycoerythrin. So this light, the blue and the red light, can't penetrate the water as easily because it's absorbed to a greater extent by the water. So green seaweeds can't photosynthesize in deep water because they can only absorb this light, blue and red, but it's being absorbed by the water instead. Our fifth mark point is then red seaweeds have less competition from green seaweeds in deeper water, because the green seaweeds are not found in deeper water because they can't photosynthesize efficiently in those deeper waters. And we could then say in shallow water, red seaweeds would have more competition from the green seaweeds. So we get this disparity where we're more likely to find red seaweeds in the deeper water because they absorb green and yellow light that is not absorbed to a great extent by the water, so it can penetrate the water. And the green seaweeds are found further up the shore where there's less water, so much shallower waters, because they only have chlorophyll. They can only absorb red and blue light, which is, as we know, absorbed by water. So if there's less water there, it means they can absorb that light for photosynthesis to be used in non-cyclic photophosphorylation. Now in this question we're actually given a diagram to start with some apparatus and some additional bits of information, and we're given a body of text. Now we're going to read through the text and highlight the key points to be able to then utilize these in our answer. Now the question says gas exchange in an aquatic plant was investigated by placing shoots into tubes containing a certain indicator called bromothymol blue, and that is a pH indicator. So firstly, we know these are aquatic plants, and aquatic means that these plants survive and live within water, and the carbon dioxide can be dissolved in the water or it can be as hydrogen carbonate ions, and that is then fixed during the Calvin cycle inside the chloroplasts of these cells from the aquatic plant. So these aquatic plants can photosynthesize within water as long as they've got a source of carbon dioxide, and they will absorb, as we know, different colors of light depending on the pigments they have inside their thylakoid membranes. Now the bromothymol blue indicator, as we just mentioned, is a pH indicator, and it gives us what color it will turn under certain conditions. So we know if the pH of this solution in each of the tubes is below pH 6, then it will go a yellow colour. It then tells us that if the pH is between 6.1 and 7.5, it will be a green colour. And it will be a blue colour in acidic conditions, pH 7.6 or above. Yeah. Then it tells us that in each of the 4 tubes, A, B, C, and D, 10 centimetre cubed of bromothymol blue solution was placed. So we know inside every tube here we've got 10 centimetre cubed of the indicator, and that is actually going to be one of the control variables whereby the same volume, 10 centimetre cubed of indicator solution, is being used. Then it says each tube was closed with a bung and left for 10 minutes. So that's another control variable that's been kept the same between each of the 4 tubes. Similar sized shoots of an aquatic plant were then placed into each of the tubes A, B, and C. Now it tells us again that this is an aquatic plant, and we can see here A, B, and C have approximately the same sized shoots of this particular species of aquatic plant. So another control variable is the species of aquatic plant that's being used. It has to be the same species. Ideally, it could even be the same shoot that's just used multiple times, one after the next. It then tells us that the 4 tubes were placed at equal distances from a 60-watt lamp and left for 1 hour. So we know that the light intensity from this lamp is going to be the same for each tube and each solution, because the tubes are placed at the same distance from this particular strength lamp. However, we are then given additional information on the actual diagram, and it does tell us that in tube A, there's nothing that's being placed around the tube. So we know that all wavelengths of light, all colours of light from the light source will be able to hit the aquatic plant. So all the different colours on the visible spectrum will be able to hit the pigments on the thylakoid membranes within the chloroplasts of these plant cells. Now the second tube, B, it says black paper has been placed around the tube. Now we know black filter paper absorbs all wavelengths of the visible spectrum. So when this light is shone at the same light intensity onto tube B, we know all the different colours of light are actually being absorbed by the black filter paper. So no wavelengths of light are actually hitting the photosynthetic pigments inside the chloroplasts of this aquatic plant. Tube C, this time, they've placed a muslin cloth around or a muslin filter around the tube. Now it tells us this lets some light through. So this means that some light will be able to hit the photosynthetic pigments, but just not quite the same amount of light as in tube A on the left hand side, because the muslin only lets some light through. Now all the different wavelengths of light will still be able to get to the photosynthetic pigments. There'll just be less of this light getting through. Now in tube D, we know there's no aquatic plant, so this is going to be our control experiment, and it's going to allow us to determine that any colour changes in the indicator are due to the activity or photosynthesis of the aquatic plant. So we know that here we've got a control experiment with no aquatic plant in, and we know no photosynthesis and no respiration are going to occur within this tube. Now we were also given a table of data along with the apparatus in the diagram. Now when we get a table of data such as this, we're going to jot down some key points next to the keywords. So we can see the initial colour of the indicator in each of the tubes A, B, C and D was actually green. So it starts out at the same colour, which indicates the same pH. Now the colour of the indicator after 1 hour in tube A, where the aquatic plant was subjected to all colours of light from the visible spectrum, the indicator turns a blue colour. Now it's important here to add in the detail from the text that we were just given. So we could write down next to this table that pH 7.6 or above will give us a blue colour, because we know that from the information we've just been given. So we're just going to jot this down next to the table. In tube B, the tube that's covered with black paper, we know the green indicator turns a yellow colour. Now again, from the information at the start of the question, if it's pH 6 or below, that means a very acidic solution. It's going to turn a yellow colour. So the solution inside, or the indicator, sorry, in tube B that was covered with black paper will turn a yellow colour, indicating a more acidic indicator solution. Now in tube C, when the tube was covered with the muslin, to limit some light getting through to the aquatic plant, so all wavelengths of light can still get through the muslin to the plant, just there's less of it getting through. We have a green colour indicator, as we had in tube A and B, and that turns, or sorry, it remains a green colour after 1 hour. Now we know from the information that green is pH 6.1 to pH 7.5, which is in between the blue colour, which is above 7.6, and the yellow colour, which is below pH 6. Now the uncovered control experiment, which was D, where we had no aquatic plant, we start green, and at the end of 1 hour we see it's also remained a green colour. So the fact that we don't get any photosynthesis or respiration because there's no aquatic plant in tube D, and the indicator remains a green colour, suggests that there's actually not much happening in tube C in relation to photosynthesis or respiration. Now the question is going to ask us about an explanation for the results that we've just seen in the table. And we're going to start with tube A. So if we look again at what's happening here, we can start to formulate an answer. And this is going to be two separate points, two separate sentences to explain why the green indicator turns blue after 1 hour. Now we know the rate of photosynthesis is going to be greater than the rate of respiration in this aquatic plant under the conditions of tube A. Now this is because a range of different wavelengths of light from the visible spectrum can be absorbed by the different pigments, the accessory pigments, and also the primary photosynthetic pigment, which is chlorophyll A. And we know if the rate of photosynthesis is greater than the rate of respiration in A, more carbon dioxide is actually going to be used up, because CO2 is fixed in photosynthesis. It's used up to make glucose eventually. And more carbon dioxide is being used than is actually going to be produced from respiration, aerobic respiration inside the mitochondria. So this means that overall, in the solution in tube A, less carbon dioxide is present because more of it is being used in respiration than is being produced. Sorry, more of it is being used in Now in this question we're going to use a molecule called DCPIP. Now DCPIP is an artificial electron acceptor, and it's used very commonly in both photosynthesis and respiration experiments. Now when DCPIP is oxidized, that means it does not have electrons bound to it, and it is a blue color. It's a blue dye. However, when DCPIP acts as an artificial electron acceptor and it accepts electrons, i.e. it becomes reduced by gaining electrons, then it becomes colorless. And we're given this information in the diagram here. So we can start to just use our highlighter pens, we can annotate it somewhat a little bit, and it says DCPIP is a blue color, it gains electrons, and we can just jot down here, we know reduction is a gain of electrons. Think about oil rig. So oxidation is loss of electrons, and reduction is gain of electrons or gain of hydrogen. So reduced DCPIP is colorless. So it's important to use this information. Now, we've then got the results of the experiment in the table below. And it says a chloroplast suspension was made by grinding fresh leaves in buffer solution and then centrifuging the mixture. And this allows the chloroplast to be separated and then extracted. The tubes were then prepared and treated in different ways. The colour of the tube contents was recorded at the start and after 15 minutes, once DCPIP had been added at the start. This information is summarised in the table. So we can take a highlighter pen again, we can start to break down the information. Now, tubes A, B and C, what's the difference between what's been placed in tubes A, B and C? So we can see here the total volume in each tube is actually 8 centimetre cubed. So that's a control variable. Now in tube A, we've got the chloroplast suspension that were isolated from the centrifuging of the ground up leaves. We've also added DCPIP, and that's the same as the contents in tube B. So we've also got chloroplasts and we've got DCPIP in tube B. Now in tube C, we do have DCPIP, the artificial electron acceptor, but we also have a buffer solution in place of the chloroplasts. So this is going to be our control experiment because there's no chloroplasts, there's only a buffer solution in its place. Now, we can then look at the treatments that have been given to these 3 tubes A, B and C. And we can see that tube A was placed in bright light. So we know we are going to get photosynthesis here because we've got intact chloroplasts. We've got visible light, white light, that has all the different colours of visible light within it. Tube B, the tube is kept in the dark. So we know there's not going to be any light dependent stage, because that requires light, and if there's no light it can't occur. So here the pigments, the accessory pigments and the primary pigment cannot absorb visible light. So that's the main difference between tube A and tube B. Now in tube C, we know there's no chloroplasts, and the tube is also placed in bright light here. That's our control experiment. So we could say here control experiment. Now, when we start to look at the data, then we can see that at the start of the experiment, the colour of the tube contents in tube A is blue-green. Now, it's written as blue-green because the chloroplasts have a green colour, because we know they reflect green light. Chlorophyll reflects green light. That's why the chloroplasts look green. And the bluey colour comes from the colour of the DCPIP indicator, which is blue. And that's the same for tube B contents. So we've got the blue colour of the DCPIP indicator, and we've got the green colour of the green light being reflected from the chloroplasts, from chlorophyll A and B. Now, the starting colour in tube C, the contents were blue, because there's no chloroplasts, so there's no green colour. There's no reflection of green light. And the DCPIP, as we know, is a blue colour. Now, after 15 minutes, what do we see in tube A? Well, we see a green colour after 15 minutes. So we can jot down quickly next to this data that the blue colour has disappeared. There's no blue colour anymore after 15 minutes in tube A. Now in tube B, we can see the colours after 15 minutes remain the same. So the blue colour has not disappeared. So we know straight away that in tube B, the DC pip has not been reduced. Because if it was reduced, that blue colour would have disappeared like it did in tube A. So we can say for tube B straight away, the blue colour is still there, the chloroplast is still there as well, and therefore DC pip has not been reduced. We know therefore that in tube A, because the blue colour does disappear, DC pip has been reduced in tube A. Now in tube C, the blue colour remains, so DC pip has not been reduced, as we might expect from our control experiment, because there are no chloroplasts. Now we can start to look at the actual questions then. And the first question is, in tube C, it states that it was used as a control, but it says explain why this control was necessary. So it's actually asking us to say why we've done this experiment, this part of the experiment with this control that we've used with no chloroplast inside. Well, we know it's always necessary to have a control experiment. Now that's very different from a control variable. Now control variable is a factor or factors that you keep the same from independent variable value to the next independent variable value. But here the control experiment, we have no chloroplasts, and that means that the DC pip will remain blue. And that tells us that it's the chloroplasts that actually are responsible for the DC pip becoming colourless, because if there's no chloroplasts, then DC pip cannot be reduced and it remains a blue colour. So no chloroplasts in tube C resulted in DC pip remaining blue, indicating that it is the chloroplasts that are responsible for DC pip becoming colourless. And we're going to see why in a minute. Now it's also important to say that the control experiment, you must conduct the experiment in exactly the same manner with exactly the same control variables as you would have done in the regular experiment. Now explain the colour of tube A after 15 minutes. So why does tube A look the way it does after 15 minutes? Well, we know with tube A, the blue-green colour has become just a green colour. So the blue colour has disappeared. Now we know DC pip is responsible for the blue colour, and if that's reduced, the blue colour disappears. So we know that DC pip must have been reduced in tube A. Now we know in photosynthesis, when there's light available at the correct intensity, photolysis will occur inside the chloroplasts. And that's really the light-activated splitting of water to form protons, oxygen gas, and 2 low-energy electrons. So electrons are provided through the photolysis, which is the light-activated splitting of water. Now we know these 2 low-energy electrons replace the 2 high-energy electrons that are released from chlorophyll A, the 2 chlorophyll A molecules, in the reaction center of photosystem 2. So really photolysis has evolved to occur to provide low energy electrons, 2 of them, to replace the high energy electrons that get excited from chlorophyll A and released from the reaction center in PS2. Then what we can say is that these released high energy electrons reduce DC pip. When they're released from photosystem 2, they reduce DC pip, and that makes the DC pip from blue to colourless. And therefore, and so to put this as the last point, only the green colour of the chloroplasts, due to the chlorophyll, is now visible, because the blue DC pip colour has disappeared, because that's been reduced. So photolysis occurs inside chloroplasts. It's the light-activated splitting of water to form protons, oxygen gas, and 2 low-energy electrons. The 2 low-energy electrons replace the 2 high-energy electrons released from the 2 chlorophyll A's in the PS2 reaction center as they're released. The two released hydrogen electrons reduce dc-pip to make it blue to colourless. And because there's no blue colour, only the green colour of the chloroplast is now visible. Now if we now look at question G at the bottom, it says the chloroplast suspension produced by centrifugation may also contain mitochondria. So they're saying that the chloroplasts in suspension that were added to each tube may also actually have mitochondria within that suspension as well. Then it says, explain the evidence from tube B that mitochondria are not responsible for reducing dc-pip. Just put that bit in there, dc-pip. So we know that mitochondria are actually the site of aerobic respiration, and that will occur in the dark as well as in the light. So tube B, there's no light because there's black paper around the tube, so it's dark, but we know aerobic respiration still occurs. Now we know mitochondria and aerobic respiration, these reactions inside the mitochondria do release hydrogen ions and electrons in reduction oxidation reactions. So oxidation is the loss of electrons from a substrate, and they're normally picked up by the coenzymes NAD or FAD. So the contents of tube B would also become green if mitochondria were responsible for reducing dc-pip. But we know because it remains a blue-green colour, it can't be the mitochondria in this case that is responsible for reducing the dc-pip. And we know in the dark, we're going to use the information again. Okay, tube B, it remains a blue-green colour.